Yep.
Yeah. Great. Hello, everyone. My name is Natalia Costa. I am a Young ISSAD Committee co-lead, and welcome to our first Young ISSAD Committee seminar series. Today's seminar is entitled Epidemiology of GroupBStrep, invasive disease in high and low income settings. Today we have as speakers Dr. Proma Paul from the London School of Hygiene and Tropical Medicine, and Dr. Lesley McGee from the Centers for Disease Control and Prevention, join us for this first event on World Prematurity Day. Well, Dr. Proma Paul is currently a research fellow and the multi-country GBS long-term outcome study coordinator. Her study looks at the developmental outcomes of survivors of invasive GBS disease in early infancy in Argentina, India, Kenya, Mozambique, and South Africa. And Dr. Lesley McGee is the team lead for the US Centers for Disease Control and Prevention's Streptococcus laboratory. And she has worked extensively in streptococcal disease surveillance, focusing on pathogen detection, antimicrobial resistance, molecular epidemiology, and evolution and genomics. Unfortunately, Dr. Proma Paul, one of the speakers, won't be able to join us today. So we have a video for her presentation.
Hi, I'm Proma. I'm sorry I can't be there in person with you, but I do want to thank Young ISSAD for giving me an opportunity to present some of our findings. I had an unexpected family thing come up. If you have any questions after this presentation, which I'm going to have to give by recording, I have my email at the end of the presentation. I will try to join the Q&A, but I'm not sure that I am going to be able to connect for that. So today, I'm going to look at I'm going to present some of our findings from neurodevelopmental outcomes of survivors of invasive GBS disease in infants. Findings, and these are going to be findings from high and low income settings. So looking at this, this just kind of gives kind of all the potential outcomes that GBS can contribute to. And in the past, the major focus had been on neonatal death. But now we are looking to really understand the burden among maternal, childbirth, and stillbirths and child outcomes, including cases of death and disability, as well as some risk factors affected on preterm birth. And this really is important when you're thinking about the full public health impact of a maternal GBS vaccine. In previous GBS burden estimates, we had an incomplete picture of childhood disability, so that's going to be the focus of today's presentation. So I'm going to present some of our findings from long-term outcomes from two studies that were published earlier this year, one in Lancet eClinical Medicine and the other in Lancet Global Health. So in a previous systematic review of GBS meningitis survivors, we estimated that moderate to severe impairment risk was about 18%. However, most of these studies were published over a decade ago. The median age of follow-up was 18 months, and therefore they were likely to miss mild impairment. And there were multiple assessment tools, over 100 in different settings, covering different
The sound just cut out for me.
Yes, for me too.
And these gaps included differences in case definition and lack of follow-up of GBS sepsis survivors, lack of longer-term cohorts, messy assessment tools that did not cover all developmental domains, and inconsistent comparability of the tools. A comparator group was often missing, which made it difficult to understand if there was any causal relationship between GBS and developmental outcomes. And there was limited data represented from low and middle income contexts. So to address this gap, we conducted two cohort studies to look at long-term outcomes related to GBS survivors. The first one was two e-cohorts in high-income countries, Denmark and the Netherlands. There we had a follow-up of children up to 20 years. These children, there were about over 22,000 children with a history of invasive GBS and over 24,000 non-exposed children to use as a comparator. For the low and middle income countries, we conducted a cohort study in 5 countries: Argentina, India, Kenya, Mozambique, and South Africa. The age of follow-up in these children were 18 months to 20 years. It was a single time point. In the high income countries, neurodevelopmental impairment was measured using ICD codes in Denmark and special education as a proxy for impairment in the Netherlands. However, in the low and middle income countries, we did use direct assessment, and neurodevelopmental impairment was measured using over 20 different tools. Although we use these different tools based on technical ability, technical capacity in each of the sites, appropriateness in terms of cultural appropriateness, as well as age. We did do some harmonizing of that measurement to make it comparable. So now looking at the data from the e-cohort study, we see that after sepsis, there is a 1.5 to 2-fold higher risk of neurodevelopmental impairment, and 5 to 10% of the survivors were affected by impairment. After meningitis, we see a 3 to 4-fold higher risk of impairment, and 20% affected, and 20% were affected. And of that, about 2/3 were moderate to severe. Now moving to the multi-country study in the low middle income context, we enrolled a total of 158 invasive GBS survivors, which included 47 meningitis and 111 sepsis. The median age of the cohort was 6 years, and about 50.8% were female. Now looking at the risk of impairment by GBS syndrome, we see that moderate to severe impairment after meningitis is about 15%, which is similar to what we have seen before. And any NDI is 46%, which is slightly more than what was previously estimated in the review, which was 32%. Based on our study, we estimated that 6% were at risk for moderate and severe impairment after GBS sepsis, and any impairment was 35%. Mild impairment was similar between meningitis and sepsis survivors. And risk of emotional and behavioral problems was 12% after meningitis and 4.5% after sepsis. And we found that the history of GBS was associated with an increased risk of moderate and severe, as well as any impairments. And this was about 1.3 to 1.7 times higher. This is looking at a summary of the different neurodevelopmental impairments that eventually went into a burden estimate. And we can see that the outcomes after meningitis are fairly similar for moderate and severe impairment, as well as for sepsis between the high and low and middle income country context. The any NDI was higher in our study in the low and middle income country context in both after meningitis and after sepsis. So in terms of what this means and next steps, invasive GBS disease was associated with a higher risk of neurodevelopmental outcomes. The risk after invasive GBS meningitis was similar to previous reviews. Risk after invasive GBS sepsis is substantial, but generally lower than meningitis. We are also looking at outcomes after meningitis in general to see the risk of increased NDI. And that is going to be published soon. There's also, what this also highlights is the need for a standardized set of assessments that are feasible and open access. And implementation research is needed to be able to follow up at-risk neonates. So as I said, these estimates went into a model to help us estimate the global burden of GBS. And what we find is that of the nearly 20 million pregnant women who are colonized with GBS, there is at least 40,000 maternal invasive GBS disease, although the increased risk of death or disability was unquantifiable due to limited data or unavailable data. We do see that there are 46,000 stillbirths associated with GBS, attributed to GBS. Therefore, that means there is at least 46,000 fetal invasive GBS disease, if not more. Although, and these fatal invasive GBS disease do have an increased risk of neurodevelopmental disability. Again, we weren't able to quantify this. Now looking at those that are looking among children, we see that there are 91,000 infant deaths, mostly neonatal, attributed GBS, and at least 390,000 infants with GBS, sepsis, and meningitis. And this includes at least 7,000 with neonatal encephalopathy. And we estimate that this means that among the survivors, about 40,000 will develop some sort of neurodevelopmental disability, moderate or severe. And then looking at preterm, we see that there is 518,000 excess preterm births that has been that's associated with GBS. But again, given we weren't able to in our studies to really look at what that means in terms of increased risk of neonatal death, or able to quantify the increased risk of neurodevelopmental disability. But it does highlight the need to really look at that even further and really understand what the increased disability is among those that are born with a preterm birth that is attributed to GBS. All right. So I will leave it here. Thank you. And again, I apologize that I can't be there in person. If you do have any questions, please don't hesitate to contact me. My email is displayed. And if you want to read more about the project and some of our additional outcomes, please follow these links. Thank you.
Yeah, we would like to invite Dr. Lesley McGee for her talk. Thank you.
Great, thank you everybody, and thanks for the opportunity to talk with you this morning and just share some of the work that we've been doing for GroupBStrep around whole genome sequencing in our team at CDC. So I'm the team lead of the Streptococcus lab at CDC, and we We are a WHO reference center, and we do a lot of reference work. We do a lot of global work, assay development, and a lot of our work has moved, like many others, to a whole genome sequencing approaches. And so I thought it would be great just to sort of share the work that we've been doing and the pipelines and bioinformatics that we've built specifically for GroupBStrep. We also, of course, are strep, so we also work very heavily in Streptococcus pneumoniae as well as GroupBStrep. So we have similar pipelines for those other two pathogens as well. Next, can I move the slides on? Oh, thank you. So just by way of background, I don't probably need to tell this audience, but Lancefield group B streptococci or strep agalactiae are gram-positive beta hemolytic bacteria that usually are in pairs or short chains. They're common colonizers of human gastrointestinal and genitourinary tracts. And it's been recognized as a common cause of disease in humans for a number of decades. It's one of the most common causes of life-threatening bacterial infections in neonates and infants. And so, that's why we have a really interesting interest in this specific streptococcal pathogen. So obviously for the neonates and infants, we separate those out into early onset disease that occurs in the first few days of life, and then late onset disease that occurs a little bit later in the child's days after birth. And late onset disease, which we've learned from many papers that have been published in the literature, is very much characterized by meningitis. We often focus on, the babies, but GBS is also a serious and quite common cause in pregnant women and also in older adults. Thanks. Next slide. Thank you. Sorry, I have all these things popping up on my screen. So CDC established guidelines which were updated several times. Guidance was first established in 1996 and then updated in 2002, and then again in 2010 for screening pregnant women at 35 to 37 weeks of gestation. And then those moms that are positive for GBS are then go for recommendation of intrapartum antibiotic prophylaxis to help reduce transmission for preventing infant disease incidence. And this is, has been continued to be used in the US. I mean, obviously IAP is not something that is used in many places worldwide. It is quite a challenging prevention strategy to implement. So obviously there's, different ways for prevention and treatment. We have the IAP, which could either be screen-based, but can also be risk-based approach for moms, who come in to deliver, who may not have had a screening. There's also obviously a risk-based approach for using IAP. Of course, there's treatment. We all have interests in antimicrobial resistance. And for GBS, it primarily remains susceptible to penicillin, although we have seen some reports in the literature around non-susceptibility to penicillin. But we do see quite high levels of resistance to erythromycin and clindamycin. And then obviously vaccination is interesting and important, and we know that the field is moving quite rapidly towards maternal vaccination. Next slide, thanks. So this slide just shows, and it's sort of older data, but I think that the lines to the right show still similar trends. And this is data coming from our US ABC's data, just showing the impact of IAP guidance in the US and seeing the dramatic decrease in early onset disease. But we see, less impact on late onset disease, and sort of over the years we have seen slight increases in that late onset disease. So the late onset disease still remains an area of interest for targeting prevention strategies. Next slide, thanks. So I talked a little bit about ABC. So folks on the line may not be familiar what ABCs is. So in the United States, we have a surveillance system called the active bacterial core surveillance. And it's a system that includes several of our states. It's 10 states within the US. And there's several pathogens that are part of the surveillance. It includes GroupBStrep, where we actually collect isolates from seven of the 10 states. We also have Group A strep, and we have strep pneumo, and then Haemophilus and Meningitis are also part of the ABC surveillance. But within our team, we focus on the 3 main streptococcal pathogens. Specifically for GroupBStrep, we receive around 2,000 to 2,500 invasive GroupBStrep annually. So these are only invasive isolates that are coming into us for testing, and these come from all ages, so infants as well as adults. And the surveillance has been in existence since 1998, and all these samples that come in get phenotypic testing done on them up until 2015. And then after 2015, we moved to a sort of whole genome sequence approach for the surveillance. Next slide, thanks. Yeah, so as I mentioned, traditionally these isolates would come in and they would have, for GBS, would have serotyping done, and then antimicrobial susceptibility was performed by broth microdilution. And that was really sort of all the data that we really did to characterize GBS up until 2015. We may have had some special studies where we may have done something like MLST, but really up till 2015, it was really just serotype and antimicrobial resistance for these pathogens. And then in 2015, we had a year in which we did side-by-side characterization of the strains. We did the phenotypic testing, and then we also did whole genome sequencing. And in that year, we developed the workflow for whole genome sequencing for these surveillance isolates. And then we also worked to develop our GroupBStrep typing pipeline. We still have one of our sites, Minnesota, still does phenotypic antimicrobial susceptibility testing. So we use that data as part of sort of a QC that we do every year. And then we do also still do within the larger surveillance some phenotypic testing on isolates where we may not have confidence or resistance might be very rare, and we want to make sure that what we're predicting from the genomic data is what we see phenotypically. So we do some phenotypic testing. Next slide, thanks. So what's in this pipeline? So the genomics pipeline, of course, pulls out the serotypes that we are interested in. From the genome, we also have scripts that will pull out multi-locus sequence typing. And then, of course, you have the whole genome data, so you can do any kind of additional SNP analysis. You could use PopBunc, you can do, any of that other phylogenetic lineage analysis that we've moved a lot more to in more recent times. Of course, we have a keen interest in antimicrobial resistance, so there are a number of key resistance and antibiotics that we focus on, and these were built really specifically for GroupBStrep. We have the pipeline that pulls data for beta-lactams. We have them for the macrolides and the lincosamides and streptogrammin group, fluoroquinolones, tetracycline, rifampin, vancomycin, and chloramphenicol. And there in parentheses, you see the different genes that we target for prediction of resistance or susceptibility. We also do use, our pipeline does also run through some of the sort of globally available AMR pipelines like ResFinder and Argonaut. So we kind of have that as sort of a little backup for anything that's maybe new and nice that may come along. I'm going to save questions till the end, if that's okay. I see a few popping up here. I'll address those at the end. Thanks. Next slide. Thank you. So in addition to, obviously the serotyping, the MLST and the AMR, we also look at some additional interesting features from the bacteria themselves. So some of the surface proteins that are highly conserved and present in almost all GBS, and some that may be of interest for vaccine development. We also have some scripts that will pull those interesting features out. One of them, of course, is the HVGA gene that we know, and people have published, really a lot in the literature more recently on this hypervirulent GBS adhesion protein that's been associated with virulence, especially in clonal complex 17, and quite highly associated with meningitis cases as well. And then we look at, obviously, the three main pili genes. And then, as I said, we look at a number of the alpha family protein, as well as the serine-rich, the SSR repeat proteins that are also important for adhesion. So just some interesting sort of adhesion proteins that have been shown to be important for carriage and disease in GBS. Next slide, thanks. So I just put up here two papers. We, like everyone, I think we all got derailed a little bit with COVID and other things that have pulled us in many directions. But the paper at the top is the paper that really sort of described a lot of the details around the pipeline. And so I've put that up there in case you have interest to go and look at that. And all the scripts are all, open source. And if anyone has interest in trying to use the pipeline for GBS, just reach out to us. I know through the Juno project with the Sanger Institute, they've taken the pipeline to use for that project. And so we're happy to work with groups, if you're doing whole genome sequencing and would like to use the pipeline. And then we did a paper a couple of years ago, and I'll be showing a little bit of that data here, where we looked at the first 3 years of our surveillance data using sort of the whole genome sequencing, and then sort of looking a little bit more into clonal complexes and some of the resistance determinants and some of the other adhesion features as well. Next slide, thanks. So here's the data here, and this sort of represents around 6,340 strains. As I mentioned, we have around 2,000 strains that we receive every year. I put in their parentheses, we now have over 14,000 sequenced, because we're now, in 2022. We have data sequenced through 2021, and so we're hoping to have some updated data published soon, and even to present at the next ISSAD meeting in Brazil. But the data is similar to other places. Of course, we have a number of we have 10 serotypes, but really there are 6 that really are predominant amongst disease. And this is, of course, is all ages here. So in the bar chart, you can see the distribution of the serotypes, and those 6 count for over 98% of samples. The interesting, just to draw your attention to there, is the serotype 3 in the orange there, which is ages 7 to 89 days, and that's your late onset disease, which, as I mentioned before, is quite commonly found within late onset disease, and is often meningitis cases as well. Next slide, thanks. So obviously, this is data just sort of showing some of the distribution of the GBS, and this is also all ages using MLST, and this is just showing you sort of the eBurst structure, and like most strep and most bacteria, it does form quite nice clonal clusters. We really see sort of 7 major CCs there listed there, and on the very left-hand side there you see the infamous CC17, which is very much found within serotype 3. You can see all the light orange color. So we do see some serotype clonal complex partnership with each other, but then you can also see that there are some CCs where you see a lot of different colors. And so in GBS, we do see a lot of examples of capsular switching within sequence types, even more so than what we see in Streptococcus pneumonia, for example. So there are a lot of examples of capsule switching. Thanks, next slide. And this is just sort of showing you the distribution of clonal complexes by age groups. In the first column there, you have early onset disease in the distribution, and then there you have late onset disease, and you can see the big gray bar in the middle again, it's our CC17. And then all other ages, you have quite a fair distribution of different CCs amongst the older age group. Thanks, next slide. And then, yeah, as I mentioned, we also look at the different pilus genes. I mean, GBS, commonly carry, just a single pili, or it can be a combination of more than one pilus gene. And so there you can see some examples of pilus biclonal complex, and you also see some quite tight associations of pili with clonal complexes as well, which is interesting. Thanks, next slide. And then similarly, the alpha proteins themselves also have a tight correlation with sequence types and clonal complexes. And so your serine-rich SSR1 is mostly, exclusively associated with serotypes 1A, 1B, 2, 5, 6, 7, 8, and 10. And then your SSR1 plus 2 together are really seen within serotypes 3 and 4. And then, as we mentioned before, the interesting HVGA, is really primarily restricted to serotype 3 in the CC17. Although we did see a few non-type 3s. We had 7 type 4s and 8 type 4s. One of them was CC452. And one was actually a CC17 type 4, so an example of a capsule switch. So you do see the HVGA really outside of serotype 3, but really mostly associated with serotype 3 and CC17. Thanks, next slide. And then AMR, we know, of course, we're always interested in antimicrobial resistance. We published a paper a few years ago, really focusing in on beta-lactam non-susceptibility, and we, while most isolates do still, retain susceptibility to beta-lactam antibiotics, we do see some changes in sort of first step changes in PBP genes that do drive resistance, up over sort of the more basal susceptibility breakpoints. And so they're not pushed over into clinical non-susceptibility yet, but we do see some sort of movements of strains creeping up a little bit with point mutations to a number of beta-lactam antibiotics. We also found within the pipeline also can look for VANG, vancomycin resistance determinants, and at least through 2017, we had 2 isolates that were confirmed to be vancomycin resistance. We see a lot of tetracycline resistance in GBS, and I know there's been some papers published around tetracycline resistance in GBS and some of the hypotheses as to why that is so high in GBS. And then for the macrolides and lincosamides, I mean, we definitely have seen within our ABCs surveillance over the years, trends towards increases in macrolides and clindamycin resistance as well. Thanks. Next slide. So this is my last slide. So obviously I was showing you data that was fairly old data. As I mentioned, we now have over 14,000 GBS genomes available to us through our surveillance system. We have a number of ongoing analyses, and we're hoping to share many of those at the next ISSAD meeting. Like many of us, we have had some delays with COVID. I think GBS is interesting. It's a little bit different from our GAS and our strep pneumo, where we have seen real declines in numbers of GAS and pneumo coming through the surveillance system. But of course, GBS is not a respiratory pathogen, and so our numbers have remained relatively high within the surveillance system. And so we're interested to look at that sort of trend data over time. So I will stop, and I'm happy to take any questions. I saw there was maybe a few, and some acknowledgements. Yeah, just like to thank our ABCs program and our Strep lab for all the work that they do in developing the data.
Thank you, Dr. Lesley. We would like to thank you for being here today and for this great and informative presentation. The CDC pipeline for GBS is indeed impressive. We have some questions, and I invite all of you to send your questions in question answer box and in the chat, or raise your hands if you wish, and open your mics to the questions. So first we have two questions from Dr. Tatiana Pinto from Brazil. First one is, did they ever have a GBS strain contradicting in serotyping and molecular typing regarding serotype? And the other one is, any GBS strain without pilus?
Yeah, so so the first one, you know, for molecular typing, we have a PCR-based approach also, and then obviously we have the ability to pull out of the genome data, but it works very well. I mean, you don't really get contradictions in serotyping between the two methods, but for sure, where you may have a difference is phenotypically you may get a non-typable, and you're actually able to type that by a molecular approach. So PCR or whole genome sequence would actually give you a serotype. So that's really the only place where you may get some difference is within the non-typables. And then the pilus so we've never looked for pili genes with a PCR-based approach. We've only used a whole genome sequence approach. And I think we did have a few strains that we did not find a pilus on, but I would have to confirm, but it's extremely rare. Most of the GBS do have pili. And that may be, you know, one would have to make sure to check that it's not, you know, a limitation of your pipeline, as opposed to actual lack of a pili gene.
Yeah.
How big is the difference between?
Yeah, we have other questions in the question answer box. So the one from Leandro Simões. Do you think that bioinformatics approaches may help us in the pursuit for a GBS vaccine? Thanks in advance.
Oh, I mean, yes, you know, I think especially, I mean, we know that the vaccines coming are all, you know, at least the most recent ones are focused on the serotypes on the capsule. So having capsular data is important, but that could come from, you know, obviously non-genomic methods. So, I mean, whole genome sequencing is a method to find serotypes. So I think it's always important to know serotype. But I think it's also as you know, vaccine companies are often looking for other vaccine targets, protein vaccines. And so I think, you know, having access to whole genome sequence data from strains all over the world would help inform whether those, you know, protein targets are in strains that you find in all places. So I think, yes, I think genomics and bioinformatics, you know, and not obviously only for GBS, but for other pathogens, I think would add important information.
Okay, thanks. So next question is from Marty Perhack. Could babies still born due to GBS also be included in the ABC surveillance?
Sorry, was that babies born?
Babies stillborn.
Oh, stillborn.
Yeah.
Yes. Oh, hold on, sorry, my phone's ringing here. Yes, I believe that ABCs does capture stillborn babies within the surveillance system, because if they have a GBS identified. So I mean the surveillance system is based on I mean, you would be a case if you have a GBS isolated from a sterile site. So if you met that definition, and obviously it would come from cases who survive or don't survive. So yes.
Okay.
I believe that is true. Yeah.
Okay. So next question is from Roberta Creti. Thanks for your very interesting presentation. I suggest to introduce the search for high-level gentamicin resistance gene and other aminoglycosides resistance genes in your pipeline. It seems to be an emergent AMR in GBS, even if clinical impact is still unknown.
So actually, yes, we do, and I did not show that here, but we do have, I think we have scripts that do look for the gentamicin and aminoglycoside resistance. And remember these, as I mentioned, the pipeline also includes the more global AMR pipelines that are out there that are not necessarily pathogen specific, but we have had some cases where we do find aminoglycoside and gentamicin resistance in GBS. That's a good point. That would be interesting to look at. Thank you for that point. I'll make a note.
Next question is from Elizabeth Martins. Do you have strains with Pilus Island 1 alone? Only Pilus Island 1?
Yes, I think so. Let me go back. Hold on, let me look at my table. I feel like in my table back here, there was, I'd have to go back to the thing, but yes, I believe we have ones that only have Pili 1. I'd have to get back to you on that. I don't know off the top of my head. Sorry.
No problem. Next question is from Stephanie Long. Do you do you carry out any sort of phenotyping?
Phenotyping, as in, can you be more
specific?
More specific, Stephanie? Do you mean phenotyping? Do we continue to do any phenotyping on the strains, if that was the question? Yes. So where we do phenotypic testing is, as I mentioned, one of our states, Minnesota, still does antimicrobial resistance susceptibility testing by broth microdilution. So we every year have, and they contribute quite a large number of isolates to the surveillance system. So it's, it is like, a couple of hundred isolates will have that phenotypic data. And then wherever we have, rare resistance, like say, for example, if the pipeline predicted, vancomycin resistance, we would then reflex and go back into the lab and make sure that we do the phenotypic testing. So for the rare, resistance, we do some repeat testing. And then we do some QC of repeat testing. A certain percentage, like 10%, gets AMR redone and serotyping redone. So yes, we do. And where we see rare serotypes also, like serotype 9 or 7 or 8, which are sort of lower, we will also confirm those as well. So yes, some phenotypic testing for sure. I hope that answered your question, Stephanie.
We have a last question from Zlotved. And how big is your difference in percentage between phenotype and genotype?
You mean, is that referring to serotype, I'm assuming? I don't know what you mean by phenotype and genotype.
Maybe the results. I don't know.
Maybe the prediction of serotype versus the genotypic. Yeah, I mean, the correlation is excellent. I mean, as I said, if it's referring to serotypes, specifically, very good correlation between serotype prediction from using whole genome sequence approach. Obviously where it is, as I mentioned before, where it is a little different, and that would be true of any molecular, whether you were using whole genome sequencing or PCR, would be around the non-typable. So you may get non-typable, if you were doing latex or Lancefield, but you could get the serotype if you did a molecular approach. And we see that a little bit more. We see we see more non-typable in GBS phenotypically than we do like in Streptococcus pneumonia, for example. But the concordance for predicting serotype is really excellent. And then, of course, for AMR, the AMR is also very good. I mean, of course, you will have some rare cases where, you know, a gene may not be, you know, it might have a mutation that doesn't make it active. And so, for example, you might predict that it's tetracycline resistance, but it's actually susceptible. So there are a few rare cases. And if you look at the publications, you know, from Ben Metcalf, the very first paper, it shows very nicely the side-by-side comparison of phenotype versus genotype.
Okay, so thanks, Dr. Lesley, for being here again, for a presentation, for answering all the questions. Now we need to move to closing remarks. So, Carol, the floor is yours.
Guys. Hi everyone, I'm Ana Caroline Botelho. I'm a postdoctoral fellow in Instituto de Microbiologia on UFRJ. And first of all, we would like to thank you, Dr. Lesley and Dr. Proma, again, for accepting our invitation. We like a lot. We would like also to thank you all for attending this first webinar and invite you to like and follow our social media, as Laura said, wrote on chat. Visit our site and blog, follow us on Twitter, Instagram, and join our group on LinkedIn. In our last part, let's say that this webinar recorded and will be available soon. Hope you enjoyed it, and until next time.
Great, thank you all. Really appreciate the chance to present and look forward to seeing you hopefully at the next ISSAD meeting. Thank you.